Good afternoon, everyone, and welcome. I'm Jack Van Horn from the University of Virginia, and it is my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and from a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across the diverse themes in biomedicine and how they relate to data science. Our selected theme for this year is focused on quantitative approaches in spatial multi-omics for guided personal medicine, where each Friday we will enjoy presentations from a wide range of leading thinkers about the issues, promise, and opportunities and hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic-based methods, along with a wide variety of data analytic opportunities they present for data science, deep learning, AI, and spatial modeling. We are pleased to announce that our 2024-2025 application cycle is open, and we hope that you or any of your junior faculty colleagues working in the various areas of omics-related biology, as well as clinical medicine and data science, will be interested in applying. Aligning with this year's theme, we are eager to receive applications from folks working in multi-omic data analytics, computational biology, machine and deep learning, spatial statistics, network analysis, graph-based metrics, as well as software design and user interfaces. Participants selected for our special Biomedical Data Science Innovation Lab program will utilize the content from lecture presentations that we're going to hear about today as essential material in a culminating in-person manuscript and grant development workshop to be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. We are very excited about this year's program where junior faculty, biomedical, and computational experts will come together to develop novel joint projects at the intersection of quantitative approaches and applications for spatial multi-omics data analytics. For today's seminar, I am delighted to welcome our speaker, Professor Audrey Hendricks from the University of Colorado's Anschutz Medical Campus. Dr. Hendricks is a tenured associate professor and alumni of the Biomedical Data Science Innovation Lab, and she's very passionate about the increased inclusion and representation in all areas of science, including those who participate in research, what questions get asked, and to whom that research is most useful for. Dr. Hendricks and her team work at the intersection of biomedical and statistical machine learning methods development with current projects, including the development of methods to increase the utility and equity of publicly available genetic resources, identifying the biological mechanisms of healthy diets, and elucidating the genomic underpinnings of conditions and traits. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Professor Hendricks' lecture today will be entitled Opportunities and Challenges for the Use of Common Controls in Sequencing Studies. Common controls provide an opportunity to increase sample size and substantially provide power to existing studies or provide controls where none exist, which can be super important for underrepresented populations or conditions. But the differences in data ascertainment and the generation of data can result in confounding and biases if not properly considered and adjusted for. Dr. Hendricks today will discuss these challenges and present solutions for robustly incorporating common controls in sequencing studies, and the considerations for newer technologies such as federated learning and implications for single cell and other omic data types will also be discussed. As always, we are streaming this lecture live and for recording via YouTube. So if you're watching on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants and alumni are encouraged to submit any questions they might have via the chat feature in their YouTube sessions. I will be synthesizing these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, thank you so very much, Audrey. We are very much looking forward to your presentation.
Wonderful. Thank you so much, Jack, for the introduction. As Jack mentioned, I'll be speaking about opportunities and challenges for the use of common controls in sequencing studies. So, as we all know, there has been amazing progress in genetics of health and disease. Are our chromosomes, and we can see our little lollipop over here on the left. And this represents an association between a region of the genome and some condition or trait. And as we start in 2005, we see just one lollipop, but as we continue up through 2010, we have hundreds, 2015, thousands, and then it just explodes, and even more so now. However, inequity continues to exist in genetics and biomedical research. And so this can be represented in part by the GWAS diversity monitor, where on the left we see participants by ancestry group, where pink is European ancestry and blue is Asian. And we can see the representation is almost all European in 2023, with some Asian and very few African, African American, Hispanic, and so on. Now, if we look on the left at all of the associations discovered, we see that even though there's an underrepresentation of participants in the African and Latinx and other groups, we can see that there's a much larger proportion of discoveries made from these groups. And this is not unique to 2023. We see this trend in 2022, 2021, 2020, 19, and so on. And so including more diversity can greatly increase genetic discoveries and improve our understanding of the genomics landscape for all people. So admixed genetic populations are underrepresented, such as African American groups, Latinx groups, and they are also underutilized in research. And this is in part because it's hard to control for substructure and to avoid confounding and bias. This propagates inequities in research and medicine, and underrepresentation occurs by other factors too, such as race, ethnicity, environments. Certain conditions and diseases are understudied. And so the propagation of inequities in research and medicine can be seen by this figure, where we have the prediction accuracy of a genetic risk score developed from the European sample, and then we have a variety of populations on the x-axis. And so we can see that the farther we get away from kind of that European type sample, the poorer the prediction accuracy. And this has been shown consistently. Representation is important. Studies of conditions that are prevalent in underrepresented groups may not be studied if we don't have those people. And again, representation gives us a better understanding of the entire genetics, genomic, environmental, and health and disease landscape for all people. And so, you might be thinking, great, well, let's just sequence everybody. And so here's the cost per human genome. And we can see the cost has been decreasing hugely and much faster than Moore's law. But even if we look at $100 per genome, which, is coming or is already here, that if we wanted to sequence 10,000 genomes, that's still $1 million. And so we know that sample sizes, very large sample sizes, 10,000, 100,000, or even more, are needed for adequate power for rare variant association studies. And examples of large sequencing data sets do exist, such as the Genome Sequencing Project, the TopMed, All of Us, the UK Biobank, among others. But most samples are actually relatively small, in the hundreds or thousands. And it's expensive to sequence controls in parallel with cases. So this means it's going to be difficult to get large representative control samples for all conditions and a large representative case sample. So a traditional case control study, we would gather our cases and our controls at the same time, and then we would generate genetic data on these cases and controls, and then we would analyze. But again, if we're looking at 10,000 cases and 10,000 controls, this is $2 million at $100 per genome, or 100,000 cases and 100,000 controls, it's 20 million. It's a lot of money. And so maybe we can incorporate external controls. And now we have our cases, and then we use externally already available data sets, such as maybe Nomad or TopMed. So we recruit and generate genetic data or sequences, or they have done these already. And then for our cases, we recruit and generate our genetic data separately and bring this together to analyze. Now, with savings, we could sequence more cases or increase diversity. So leveraging existing external common controls has the potential to increase power for relatively small studies, provide controls where none exist, for example, secondary analysis of germline variation for cancer cases, and to free up resources to sequence additional cases, perhaps for uncommon conditions, more diverse samples. And so if we look at a Nature review paper where we investigated this, we thought a lot about where might we want to incorporate external common controls. And so on the y-axis, we have our genetic architecture where we're thinking about conditions that might be more Mendelian, caused by just one or a few genes, and then up through polygenic caused by a large number of genes. And then on the x-axis, we have condition prevalence. So how common is the condition? Is it very rare? Is it uncommon? Or is it more common? And so we can see that if we have conditions that are caused by one or a small groups of genes and are very, very rare, that we have existing analysis and study design methods for this, such as small family studies or case groups. And on the other extreme, for very large control studies that are highly polygenic, we can garner huge sample sizes. So maybe type 2 diabetes, autism, coronary heart disease, and so on. But somewhere in the middle, we have conditions that are not particularly rare, but they're uncommon. Maybe they only occur in 1 out of 100 people or 1 out of 1,000 people. And they are more polygenic. How do we garner a large enough sample size to understand the genetic architecture of these conditions? And so this is where we think common controls can be especially valuable. And so common controls have been used in a variety of conditions and papers. But incorporating external controls can cause confounding and bias, or false positives and false negatives, because again, we are recruiting and generating our data separately for our cases from our controls. And so our challenges, I'm going to group into three groups. One is we can have ascertainment differences between our cases and our external common controls. We can have population structure, genetic similarity differences between our cases and external controls, and we can have technology and processing differences. And so we've thought about this a lot with a variety of colleagues, and we want to think about how we can better improve the incorporation of external common controls in the pre-analysis. So we want to start with our research question and which external common controls do we include? Do we include individual data or summary data? What type of ancestry background are we including, and so on? We also want to think about our harmonization and analysis pipeline. Can we filter out or control for depth of coverage or genotype quality? And of course, we want to be extra careful with our results. We might have additional verification that we can do, potentially in silico validation, looking back at depth of coverage and so on. Partial replication, maybe we incorporate another external control group. And then we want to make sure we're being careful with our interpretation. So a lot of my work is focused on summary level external controls. And so when we're thinking about individual level data or summary level data, for individual, the pros are that we can more easily detect and control for differences between our internal and external samples. But the cons are that individual level data are often more difficult to obtain and are more computationally intensive. For summary level data, they're often easier to obtain and have fewer computational resources needed, but it's harder to detect and control for differences between your internal case sample and your external control sample. And so if we look at the resources available here, individual level on the left and summary level on the right, we can see that often the individual level data has fewer permissions required. The individual level data often has more permissions required than the summary level data. And the summary level data is often much, much larger than the individual level data. Additionally, the use of genomic summary data is poised to continue to grow. We have a lot of hospital-based biobanks with genetic data, increases in other genomic summary data types, such as methylation or expression, and new technologies such as federated learning, where we're not sharing individual level data across different sites here represented by our local nodes. Instead, we're sharing summary level data back to a central model or server where we combine everything. And so our summary data is going to be included and used regardless of whether we have individual level data to start. So how do we work to address the problem? We develop methods, recommendations, and software to increase the utility and equity of genomic summary data. We work to enable rigorous and reproducible research for all samples and to make our methods and software easy to use. So I want to start over here with differences in technology and processing. And so this work has been led quite a bit by Jessica Murphy, a PhD student in our team. And so on the left here, we can see that differences in coverage or technology can produce differences in where and which variants are detected. So high depth whole genome will detect perhaps the most number of variants. High depth exome will only detect the variants within the exons, and low depth genome will sporadically detect variants throughout. Additionally, we can have differences in processing or computational pipeline where we can see that the variants we detect will differ. So we worked and developed proxy external controls association test to help address this, where we start by thinking about a traditional case control burden test, where we have under the null hypothesis of no relationship between case and control status and our rare variants in a gene, we would expect the rate of rare alternate alleles in our cases for our gene of interest to be equal to our rate of alternate alleles in our controls. Now, proxy external controls association test, ProxyCat, takes this and weights now by a proxy. So you can see the numerators are exactly the same as what we have up here, but now we have a proxy in our denominator. Now, it turns out that if we assume Poisson distribution, that the maximum likelihood estimates have a closed form, and all this means is that we have a very, very fast test. And so then our question is, what do we want to use as our proxy? Well, we would like our proxy to capture our ability to detect rare alternate alleles in each gene region. We could use depth of coverage in that gene region. We could also use rare non-functional, or in other words, synonymous variants. And we chose to use the latter because quite often when we analyze rare variants for genetic association studies, we throw out the non-functional or synonymous variants, but that data comes along for the ride regardless. And so we can see this here where we have a variety of genes and we have our cases up top and our controls below. And we have rare synonymous variants represented by the black dots and the rare functional variants represented by the stars. And so we could look at gene 2, where we can see quite a few rare functional variants, and additionally more in the cases and the controls, but we see the same number, approximately the same number of rare synonymous variants. So this might be a gene that is actually associated with case status. Alternatively, in gene 3, while we also see a higher number of rare functional variants in our cases versus controls. We also see a higher number of rare synonymous variants in our cases and controls. And this suggests that we just might be sequencing or detecting variants better in our cases than in our controls, perhaps due to technology or pipeline differences. And we think that this could be an example of a confounded association. And so coming back to our equation under the null hypothesis, now we're looking at the alternate allele rate in our cases for functional variants over synonymous variants in our cases. And again, this same ratio in controls. This controls for differential gene bias in cases and controls. And so we looked at our type 1 error rate where we had no association and no confounding, and then over a variety of confounding scenarios where we had gene level confounding in cases and controls or only cases, where we have case control confounding, and then where we also have both gene and case control confounding. And we compare ProxyCat in blue to a traditional case control likelihood ratio test in black, and then also a case only test where we're comparing to the average across the genome. And we can see that ProxyCat is the only test that maintains the appropriate type 1 error. We applied this using the Severe Childhood Onset Obesity Project cases. These are whole exome sequenced at a pretty high depth of 60x. And then we used exact, the nomad precursor of about 30,000 non-Finnish European, whole exome sequenced at about 10x. And we can see that the traditional likelihood ratio test in black is quite inflated, both at the median and also in the tail, but ProxyCat and a ProxyCat weighted, which further adjusts for differences in sequencing depth, are much better controlled. And so this paper came out in 2018. Now we've since worked on expanding ProxyCat into a logistic regression framework, and we're excited to move forward with that. I'll show you a little bit of that later in the presentation. So now I want to go on to controlling for ancestry or population structure. So when we think about opportunities for genetic summary data, we might use genome-wide association studies from the GWAS catalog or the Pan-UK Biobank. There's other public large genetic databases available, such as allele frequencies from Nomad or Alpha through dbSNP, and large sequencing programs such as TopMed and GSP. Biobanks such as the UK Biobank and All of Us. And again, it's often easier to access and use compared to individual level data. And genetic summary data is used for many analyses, such as polygenic risk scores, causal inference through Mendelian randomization, or genetic correlation. Also used to prioritize putative rare causal variants, and as external common controls for case control association tests. But if we look at some of the data we might consider using as external common controls, a lot of this data has substructure. So here I'm representing some groups from the Nomad aggregation database, and version 4 has over 700,000 exomes and over 70,000 genomes, so it's quite large resource. But if we look at some of these groups, we can see that we would expect them to contain some genetic ancestry substructure. For the American Latinx, we might expect European, Native American, and African, and for other, maybe a little bit of everything. So our motivation here is that incomplete genetic ancestry information can result in incorrect analyses and conclusions about genetic associations and incorrect prioritization of putative causal variants. And other people have talked about this as well, and we are going to use this paper right here as a motivating example. So if we look at this paper, It is studying severe hair loss in women of African ancestry. This is arguably a very important question in an understudied condition, an underrepresented group. And if we look at the statistical analysis that was used, we see that the only control set that was used was the African-American ancestry group from Nomad. Now, I would argue that this is a suboptimal and inequitable choice because the authors either have the choice to not use the resource at all or to use the best but poorly matched external data set. And the sheer existence of this choice exacerbates inequities because it's not a choice that we would need to make for a non-admixed group. So our motivation is to estimate genetic similarity substructure in allele frequency data so that we and others can robustly use all genetic summary data, including admixed data. But first, I want to take a step back and talk about this wonderful study in National Academy's report that came out in 2023. And hold on, I'm going to put down my blind because it's getting very sensitive. Okay, thank you. So if you haven't seen this report, I strongly recommend that you take a look. A few weeks ago, another National Academy's report came out about race and ethnicity that is also quite good. So in this report, the authors really thought deeply about these definitions, and they defined ancestry as a person's origin or descent, ethnicity as a socio-politically constructed based on perceived cultural similarities or heritage, race as a socio-politically constructed for classifying based on subjective beliefs about perceived biological similarities. And then they introduce a concept called genetic similarity, which is the quantitative measure of shared genetic ancestry. So you'll hear me using genetic similarity quite a bit throughout the rest of this presentation. Now, one of the main recommendations from this National Academy's report was that we should avoid using ancestry or other groupings in genetics and genomics research usually and whenever possible, and that it would be helpful to use genetic similarity to control for confounding. Now, with individual level data, using genetic similarity to control for confounding is quite common. We can use principal component analysis or a random effect term in a linear mixed effects regression. But for summary data, the concept of using genetic similarity hasn't been used quite as much when this report came out. Now, Nomad has more recently really thought about this deeply too. So here's a genetic ancestry blog that they wrote. I recommend reading it. And they've also started to think about the differences between ancestry, race, and ethnicity. And so here we have what Nomad looks like from Exact to Nomad version 2, Nomad version 3, Nomad version 4. And whereas you can see that we do start to have maybe larger numbers of admixed American, shown in red, and African American groups in purple, we're still predominantly European. And again, like I said, they have been thinking very carefully about how to present their data. And we're just show some of the groups here where they're also now providing study provided labels. That includes self-report or researcher assigned, as well as self-reported study provided labels that are less common. And so what I really want to point out is that many of these groups, especially admixed American and African American, are very, very diverse in terms of the self-report or researcher assigned, as well as, and no label is provided in the majority of these, as well as the ancestry, as we'll see later. So again, we wanted to address the issue that estimating genetic similarity has been quite difficult or impossible in genetic summary data. And so we thought that we could start to do this. So we developed the method SumMix, where if you have your target sample or person, you can estimate the substructure using some kind of existing technique such as admixture or so on. And then for the observed summary data, SUMICS can estimate the substructure proportions in that data and then adjust those substructure proportions to match your target sample or person, making the comparison between these two more robust. And we do this using a mixture model where we are estimating our ancestry proportions or our genetic similarity to our reference groups, K reference groups here, using our reference data, allele frequencies, and our observed data, allele frequencies. And we find that we can solve this pretty easily using least squares. And since we have some additional constraints, we use sequential quadratic programming to solve. So we applied this to Nomad version 2. And as you might expect, the African-American sample we found contains quite a bit of African-like ancestry as well as European-like ancestry. And all of these are similarity to 1,000 genomes reference groups. For the American or Latino group, we see that we have a high proportion of European-like or Indigenous American-like, and then moderate proportions of African-like and East Asian-like. And for the other group, we find that we have a high proportion of European-like and a little bit of everything else, as we might expect. Now, the other three groups that we looked at in Nomad version 2, non-Finnish European was very similar to European-like, and East Asian was very similar to East Asian-like. And while we found a high proportion of South Asian was similar to South Asian-like, we also have a moderate proportion of European-like, which is actually expected due to an ancient admixture event. So then we wanted to look at our ability to complete allele frequency adjustment and also to complete harmonization using SUMICS. And so here we're thinking, what if we have 3 different summary data sets? We can estimate the genetic similarity proportions, and then we can harmonize these data sets. And so we did this with Nomad, where we have our African-American on the left, and then we adjusted the allele frequencies to be about 100% African. And what I'm showing below is either the absolute difference in the upper plot or the relative difference in the lower. And purple is unadjusted allele frequencies. Orange is adjusted using Nomad as reference, and blue is adjusted using 1,000 genomes as reference. Now we want to be closer to 0 because that means that our data sets are better harmonized. And what we see here is that both adjusted methods outperform the unadjusted quite a bit. And there doesn't really seem to be much of a difference between whether we use Nomad data as reference or 1,000 genomes data as reference. And so we wanted to go back to our motivating example, and we reanalyzed this data. And so we calculated the unadjusted allele frequencies for Nomad version 2.1 African, and then we adjusted the allele frequencies to be 100% African. And we repeated the 2 tests that were performed in the paper. And what we can see, and should come to no surprise, is that we get different results when we adjust for genetic similarity or genetic ancestry differences. Now, it's worth noting that we don't actually know what the ancestry proportions were for the cases. That was not reported. But we do know that the cases included women from Africa and also African-American women from the US. So we suspect that the proportion is probably somewhere in between the unadjusted African-American proportions and 100% African. And if we knew the actual proportions we could adjust exactly. And so since the original SumX publication came out, we have been extending the method's capability, and this work has been led quite a bit by Adele Price and Haley Wolf in our team. And so we have extended beyond adjusting or estimating continental genetic similarity to more finer scale. And also we can estimate the genetic similarity to conditions beyond ancestry or beyond genetic ancestry, such as genetic predisposition to cases and controls. We extended SumX to do a local scan of estimation across the genome. And then we can use these genetic similarity estimates to complete a selection scan. And of course, we can use all of our new genetic similarity estimation to improve our adjustment or harmonization of finer scale groupings and also adjust locally. So I'll start by looking a little bit at the global fine scale. So previously, we looked at our continental ancestry group shown here, and now we've extended out into the finer scale groups represented. This is harmonized data with NOMAD version 3 through the Human Genome Diversity Project in 1,000 Genomes. And it's worth really reminding everyone that while this finer scale ancestry is certainly better, we do have underrepresentation in African, Indigenous American, and South Asian. We are certainly not capturing the full genetic landscape there. And so first, we find that our bias is captured by highly similar reference groups. So if we take a look at this figure where we have our SumX estimate minus our true proportion, so we're essentially looking at our bias in our estimates, we can see that we have up to about 3% bias in some of these estimates, which is quite a bit larger than what we had for the continental. Continental was within 0.1%, so very, very tight estimates. And if we look at where this bias is distributed, we see bias in African-like and European-like when we're estimating the genetic similarity to African-American-like groups. We can see that the bias is given to other reference groups that are similar. And what I mean by that is this bias seen in Yoruban down here of about 3% is distributed to other African-like finer scale groupings, meaning that this bias is distributed what we would call locally to other genetically similar groups. And it turns out that because this bias is captured by highly similar reference groups, that the bias is not propagated to our allele frequency adjustment or our harmonization. And so what we see here is we are looking at the absolute difference between the target and the adjusted allele frequencies by different minor allele frequency groupings on the x-axis. And we have unadjusted in gray, continental adjustment, so that would be the original SumX in blue, finer scale estimate adjustment in green, and then our gold standard finer scale parameter adjustment in teal. We never know actually the true parameter, so this is kind of our gold standard, but unknown. And what we see here is, again, we see that the continental adjustment is better than the unadjusted, which we saw before. Finer scale is even better than the continental adjustment. And interestingly, the estimates, even though those estimates have bias, as we saw on the previous slide, that bias is not propagated because we see the estimates are pretty close to our true unbiased parameters. And again, we believe that's because the bias is distributed locally to other genetically similar groups. So now I want to talk a bit about SumX local. So if we look at kind of a toy representation of chromosomes for different people, we can think of a local substructure block across individuals like this, and we can summarize that local substructure into proportions. So this might be what our summary data looks like. And then if we did this scan across a chromosome, we would get ancestry or genetic similarity proportions across a chromosome. And if we see a region where the genetic similarity proportions differ from the average, this could be a possible region of selection. So we wanted to think about how to detect local substructure differences. So we want to test whether a certain block, represented by W, for each kind of ancestry group, reference group, is equal to the mean across a chromosome. And we put this into a traditional walled test statistic where we're comparing our estimate to our mean over an estimate of our error. However, the biggest question is, how do we determine our standard error? Because we do not have individual level data. Again, we're only using allele frequency. And so we decided to account for our standard error by accounting for two sources of variability. One is our within block variability and a between block variability. So to estimate the within block variability, we use within block simulations to create an empirical distribution from which we calculate the standard deviation. And then the between block variability, we calculate the standard deviation across the chromosome. And then we combine these to get our standard error estimate. We evaluated the type 1 error and power of this. So type 1 error is at the top and power is at the bottom. We did this using two algorithms, a fast catch-up algorithm, which is more flexible in terms of start and end site of those windows, and then a preset sliding window approach across the genome. And we investigated a variety of parameters. Our default parameters are here, and then we would estimate smaller or larger parameters. And so what we see, first of all, is that the type 1 error is almost always well below our expected threshold of 0.05, except when we start having very small sample sizes for our observed sample or for our reference sample of about 100. Additionally, our power is also quite high, except for some of the smaller parameters. And I'll point out that the difference that we're looking at here, the change magnitude is only 3% for our default. So it's not enormous. That's the proportion difference that we would expect in that window. So we applied this to Nomad. And we have our Manhattan plot here with a negative log 10 p-value for the y-axis and then our chromosomes. And we applied this to African, African-American, as well as American Latino. And we can see that we have some replicated signals such as the HLA, which is a region well known to be under selection, as well as some potentially new areas of selection. So now I want to talk about ascertainment differences. I alluded to the fact that we can actually detect genetic similarity to any genetic grouping, such as cases and controls. So we looked into this and defined our case control study that from a polygenic risk score study. And we're trying to estimate the predisposition to disease. So are certain people predisposed to have the disease or not have the disease? And then again, we might want to harmonize this with omics to adjust for ascertainment differences. And so we looked at this in prostate cancer using 161 polygenic risk score variants in the Colorado Biobank. We use omics to calculate the proportion of genetic predisposition to non-prostate cancer controls and prostate cancer cases. And then we looked at CCPM, prostate cancer males, all males and all females. And so first, when we're looking at our prostate cancer males, where we know that all of these men have prostate cancer, we expect that we would be estimating near 100% genetic predisposition. Now, we had fewer than 10 individuals for this first group, so we were unable to get estimates here. And we get very close to 100% with our 40 to 60-year age, and we get a little under for our greater than 60-year-old age, which we believe might be due to more of an environmental contribution to this group. When we look here at all CCPM males, again by age group, less than 40, 40 to 60, and greater than 60 years old, we see, as we expect, that the observed prostate cancer proportion increases. Typically, men are diagnosed with prostate cancer as they get older, but what we didn't expect to see was that our genetic predisposition to prostate cancer also increases. You're born with your genetics, so they should not be changing with age. And what we believe is happening here is that we have a hospital-based biobank, and so men are coming in to be treated for prostate cancer, more men as they get older. Those men are being subsequently recruited into the biobank. And so this, we believe, represents an ascertainment bias. And then as kind of a as another kind of positive control, we looked at our CCPM females, none of whom have prostate cancer. They don't have prostates, and we see more of a less of an increase over age, so more consistent. Now, you might be asking, can we combine ProxyCat and Summix, because then we could control for differences in data recruitment and processing as well as ancestry? And so we were wondering that as well. Can Summix adjust for population structure and external common control methods for rare variant association tests? And so just very briefly, Summix, ProxyCat is not the only rare variant association tests. There are those that use individual level data, and there are those that use summary level data. Most notably, ICAT-O is a very interesting test that is an optimal combination of burden and variance that we'll compare as well. It's worth noting that no summary level test includes covariates, and no summary level test can include more than one external sample, and of course, none explicitly adjust for ancestry. So Sage Sigler led this work, and she wanted to ask, does adjusting minor allele counts using Summix control for confounding? And how large should the reference sample sizes be for adjusting very rare variants? So if we look at the effect of reference sample size on type 1 error for cases and external common controls, we can see first that if we use the sample sizes from 1,000 genomes, where we have about 700 African samples and about 650 European samples, we have large inflation of our type 1 error. We are not well controlled, regardless of the method that we use. As we increase the sample size that we see here for our reference data to 2,000 African and European, we have much better controlled. And again, we're looking at adjusted in the closed circles here and unadjusted in the open circles, and that's adjusting using Summix. And then if we look at 10,000 African and 10,000 European, we have very well controlled type 1 error across all of the genes that we evaluated. Of note, it is possible to get large enough reference sample sizes because we can use NOMAD as these adjustments, and we can actually adjust the African American NOMAD to be 100% African and then use that as a reference data set. So then we wanted to look at the effect of reference sample sizes when we have both cases and internal controls as well as external controls. So previously, we only had internal cases and external common controls. And so here we're looking at two methods, or a logistic ProxyCat, which I alluded to earlier, and ICAT-O. And here we don't see much of a problem actually with these smaller reference sample sizes, pretty well controlled type 1 error throughout. And so for the external common controls only method, we see inflated type 1 error with small reference sample sizes. So we need a large enough reference sample size to use these adjustments. For the internal controls plus external controls, we have a well-controlled type 1 error. So I just want to take a step back from all of my work and talk a little bit about how we might use this in single cell work. So there is a nice paper that came out, review paper, and there is a ton of deconvolution methods that exist for single cell data. And so I encourage you to take a look at this paper here. And they also went through and talked about the two general approaches for deconvolution in single cell. One is an enrichment score approach, and the other general approach is more regression-based, as well as where we might want to use deconvolution. And again, deconvolution of bulk data using single cell data as a reference can be extremely valuable and help the amazing information from single cell kind of go further into larger data sets. And I'm sure a lot of you, please take a look at my slides. These are other papers that are quite interesting from methylation and then inferring ancestry in single cell data. But I just wanted to end with, I'm sure a lot of you saw that the Human Cell Atlas came out in Nature on Wednesday with quite a few really interesting papers, as well as a data portal here and some kind of interactive activities. So I think there is a lot of opportunity here to use consortia and data from large projects such as the Human Cell Atlas for deconvolution back out into larger bulk data sets. So in summary, we can incorporate external common controls to boost power, and we can do this using ProxyCat. We can also estimate and adjust for population structure, genetic similarity. And to ensure robust and reproducible work, we must adjust for differences in data generation, technology and computation, and other sources of substructure, such as ancestry and ascertainment. This will help increase the utility and equity of summary genetic data, and it will help with additional uses such as federated learning and single cell. So I will end there. Thank you.
Audrey, thank you so much. I was just kind of thinking about all the different, sources of where these data come from, and they are presumably no single laboratory produces all of these. These are done through large-scale consortia. Hopefully, there's some sort of standards and references that they utilize, and there's probably quality control standards they have to meet. How do they wrangle all of that? Because surely there are differences between just site to site variation of who's doing the actual sequencing. Different vendors are contributing. Of course, there's technological developments. You wait 18 months and there's another way to do it. How is all that kind of managed, maintained, and is there versioning that accounts for that?
Yeah, it's a great question. So I think within certain studies, there's most certainly versioning. So I'm thinking, for instance, about the TopMed study, right? So there's certain releases that come out. So if you want to work with that data, there might be some samples that are included in version 2 and some that are included in version 3 and version 4. And of course, within the data within these larger samples, and consortia, they will have certain harmonization techniques that they'll try to use to ensure that there's not severe batch effects. Now, all of that is to say that for better or for worse, some of the study designs of these are less than optimal. And I think you alluded to this, Jack. For instance, often, even within the same large consortia, there are multiple centers for data generation. And sometimes the cases and controls might be split by data generation center. And so that could lead to potential batch effects, which can be really problematic. So again, kind of coming back to these methods where we can try to detect differences within the data and adjust for them. And then, of course, if we're trying to combine studies such as from TopMed and maybe the Genome Sequencing Program or the UK Biobank, there's going to be even larger differences because there's not as much effort put into harmonizing all of those data sets. Does that answer your question?
Yeah, it does. Um, Also, how big is big enough? You mentioned a variety of different sources. You have the 1,000 genomes, but then we have the Nomad one that has like 70,000, and there was several others that you mentioned. How many is enough, or is representative sampling, is that sufficient for what we want to do? But is it just sheer numbers? Because again, you wait a while and there'll be some other one that comes out, which is not the 1,000 genomes, but the 100,000 genomes or the 1 million genomes or whatever is next.
It's true. We do have 1 million genomes coming down the pike. I think there's one in England and the UK that's coming. Um, so, I mean, I think it's a really good question. And I think it dives into, well, one, how large is the effect that we're trying to detect? Because there are effects that are smaller, but are biologically relevant and have led to kind of drug development. That have only been captured with very, very large sample sizes. In those cases, it's quite often in the hundreds of thousands. And then if we want to try to assess different genetic or even other omic variation that we might not see in a European white background, then we are going to need to garner some of those larger sample sizes there. Now, I don't think we should wait until we have the million to complete this research. I think that there's a lot we can do in the meantime to use the data sets we have to uncover this. And also, it would be, I think, um, not optimal to also not use the current data that we have. So we are accumulating more and more data. So how do we use the data that has substructure like ancestry or population structure, like ascertainment differences? How do we try to use the data that we have to have large enough sample sizes while controlling for differences that could cause false positives or false negatives? I always like to say false negatives too, because a lot of the time we think, oh, it's only going to be false positives that we're worried about. But that's not actually true. We can be missing signals as well.
One other thing, as you were talking about genetic similarity and how you attempt to address that, that seems to be a question of kind of spatial resolution, if you will. And you sort of addressed this when you were talking about the continental related levels of similarity, or those that are more like regionally based, et cetera, and trying to, kind of clump groups into various kind of catchments, if you will. I have another question about why you don't like the Finnish, or why geneticists don't like the Finns, but that's another one. But the thing I also was thinking about is that this is a measure of spatial resolution, but it's also a measure of temporal resolution, right? The emergence of genetic material from out of Africa, if you will. We're all Africans by genetic ancestry. Eventually, you can trace our ancestry back that far. Is there any attempt to take into account that genetic flow that happens over time as different populations have migrated out of Africa and throughout the rest of the world?
That's a really, really great question. And I like how you're kind of bringing it back to kind of spatial resolution. And I will just say that we also have a measure of goodness of fit for how as a measure of how good is your reference panel for estimating the substructure in your data. And so we can tell whether it's not very good and you're missing an important reference group or reference substructure, or like you might say, Jack, you have insufficient spatial kind of depth. Um, those plots that I was showing, the network plots, the lines in between show how genetically similar they are to the reference groups are to each other. So a wider line means more genetically similar. And we use FST as an estimate of that, which estimates the genetic similarity and also to some degree also captures the kind of historical kind of migration, because you see things like African groups being less similar to other groups because we know they diverged earlier, and things like South Asian and Europeans being more genetically similar, again, because there was an ancient admixture event. So to kind of a broader or on average way, we are capturing that a little bit in the genetic similarity measures.
I think that's a really fascinating thing to try to think about. That is that if you can not only understand where that particular level of spatial resolution, kind of how you consider that, but also how people, as their genetic material has moved from person to person to person throughout their ancestry, and it is also moved with them around the world, that even though you go and group people spatially sometimes, that their actual temporal resolution is something which would be kind of interesting to kind of map in term, if you could get an estimate of just natural mutation rates. And it's almost like a biological clock, right? And you could like trace back people who have similar genomes, but also similar kind of points in time where their genomes might have diverged from each other. And maybe that event is something which gave rise to some disease states.
Yeah, it's a really interesting point. I agree.
Well, Audrey Hendricks from University of Colorado, thank you so much for sharing this fantastic talk with us. You really got me thinking. I really appreciate that. This is really wonderful. And thanks so much for sharing your work and your tools and the work of your lab. It's really fascinating work. We will not be meeting next Friday because of the Thanksgiving holiday here in the United States, but I want to wish everyone a happy Thanksgiving wherever you are. And we'll look forward to talking to you again soon. And Audrey, once again, thank you so much.
Thank you.